Human genetics, polygenic inheritance. ChatGPT said. Human genetics, polygenic inheritance 1. Introduction to polygenic inheritance. Polygenic inheritance refers to the transmission of traits that are controlled by multiple genes, polygens, each contributing a small additive effect. Unlike Mendelian traits, which follow simple dominant recessive patterns, polygenic traits show continuous variation and do not exhibit distinct phenotypic classes. This form of inheritance is responsible for many complex human traits, such as height, skin color, intelligence, and susceptibility to diseases. 2. Characteristics of polygenic inheritance. Multiple genes influence a trait. Unlike single-gene monogenic traits, polygenic traits are determined by the cumulative effect of multiple genes, often located on different chromosomes. Additive effect. Each contributing allele adds to the overall expression of the trait. For instance, in skin color determination, more melanin-producing alleles result in darker pigmentation. Continuous variation. Polygenic traits do not exhibit distinct categories but instead show a range of phenotypic expressions, forming a bell-shaped curve in a population. Influence of environment. While genes play a major role, environmental factors such as nutrition, lifestyle, and exposure to sunlight can modify the expression of polygenic traits. Quantitative traits. Polygenic traits are often measured quantitatively, e.g., height in centimeters or IQ scores, rather than classified into distinct groups. 3. Examples of polygenic inheritance in humans. Height, governed by multiple genes, each contributing to overall stature. Environmental factors like nutrition and health during growth years also play a role. Skin color, controlled by several genes affecting melanin production. More dominant alleles result in darker skin, whereas recessive alleles contribute to lighter skin. Eye color. Although previously thought to follow simple Mendelian inheritance, research indicates that multiple genes contribute to the variations in eye color. Intelligence, a polygenic trait influenced by genetic and environmental factors, with studies identifying numerous genes contributing to cognitive ability. Susceptibility to diseases. Conditions like diabetes, hypertension, schizophrenia, and heart disease have polygenic bases, meaning multiple genetic factors influence their development. 4. Polygenic inheritance in the bell curve distribution. Polygenic traits often follow a normal distribution in a population, where most individuals exhibit an intermediate phenotype, while fewer individuals display extreme expressions. This is represented graphically as a bell-shaped curve. 5. Difference between polygenic inheritance and Mendelian inheritance feature. Polygenic inheritance, Mendelian inheritance, number of genes, multiple genes, single gene, phenotypic variation, continuous, e.g., height, discrete, e.g., blood type, effect of environment, strongly influence, less influence, inheritance pattern, additive effect of alleles, dominant, recessive, or codominant. 6. Genetic analysis of polygenic traits. Since polygenic traits do not follow simple Mendelian ratios, their inheritance is studied using statistical and biometric methods. Techniques such as quantitative trait loci (QTL) mapping help identify specific genes associated with polygenic traits. Genome-wide association studies (GWAS) also aid in understanding the genetic basis of complex traits. Solve previous year questions from PhD entrance exams. 1. Question. Which of the following characteristics is not associated with polygenic inheritance? A. Continuous variation in phenotype. B. Influence of multiple genes. C. Distinct Mendelian ratios. D. Strong environmental influence. Answer. C. Distinct Mendelian ratios. Polygenic traits do not follow simple Mendelian ratios but rather show continuous variation due to the involvement of multiple genes. 2. Question. Height in humans follows a polygenic mode of inheritance. If two parents of average height have a child who is significantly taller than them, what could be the reason? A. Mutation in a single dominant gene. B. Accumulation of dominant height increasing alleles. C. X-linked inheritance. D. Complete dominance of a single gene. Answer. B. Accumulation of dominant height increasing alleles. Polygenic traits result from the additive effects of multiple alleles. If a child inherits a higher number of dominant height increasing alleles than the parents, they may be taller. Multiple choice questions MCQs for practice 1. Which of the following traits is an example of polygenic inheritance? A. ABO blood group. B. Cystic fibrosis. C. Human height. D. Color blindness. Answer. C. Human height. 2. In polygenic inheritance, the phenotypic expression follows. A single gene Mendelian ratio. B. A normal distribution curve. C. Complete dominance. D. Codominance. Answer. B. A normal distribution curve. 3. Which genetic analysis method is most commonly used to study polygenic inheritance? A. Punnett square analysis. B. Pedigree analysis. C. Genome-wide association studies. GWAS. D. Test cross. Answer. C. Genome-wide association studies. GWAS. Conclusion. Polygenic inheritance is fundamental in understanding human traits, as it explains the continuous variation seen in complex characteristics. Unlike Mendelian inheritance, polygenic traits are influenced by multiple genes and environmental factors, making their inheritance more intricate. Advances in genetic research, including GWAS and QTL mapping, have significantly contributed to unraveling the genetic architecture of these traits. Understanding polygenic inheritance is crucial in fields like medical genetics, evolutionary biology, and personalized medicine. You said. Heritability and its measurements. ChatGPT said. Heritability and its measurements. 1. Introduction to heritability. Heritability is a measure of how much of the variation in a particular trait within a population is due to genetic differences among individuals. It is a key concept in quantitative genetics and is commonly used in studies of evolution, behavior, and medical genetics. Heritability does not indicate the degree to which a trait is inherited by an individual but rather the proportion of variation in a population that can be attributed to genetic factors. 2. Types of heritability. Broad sense heritability H squared represents the total genetic contribution to phenotypic variation. Includes all genetic effects, additive, dominance, and epistatic interaction among genes. Formula. H 2 equals VGVP H 2 equals VP VG where V GVG equals total genetic variance V PVP equals total phenotypic variance genetic plus environmental variance narrow sense heritability H squared. Measures only the proportion of variation due to additive genetic effects which can be passed to offspring. Important for predicting response to selection in breeding and evolution. Formula. H2 equals VAVP H2 equals VP VA where VAVA equals additive genetic variance V PVP equals total phenotypic variance. Narrow sense heritability is more relevant in evolutionary biology and artificial selection because additive genetic variation determines how much a trait can respond to natural or artificial selection. 3. Measuring heritability. Heritability is estimated using various methods, including twin studies, family studies, and regression analyses. Twin studies. Monozygotic MZ twins share 100% of their genes, whereas dizygotic DZ twins share about 50%. If a trait is highly heritable, MZ twins should be more similar than DZ twins. Heritability estimate. H. 2 equals to RMZ minus RDZ H2 equals to RMZ minus RDZ, where R is the correlation coefficient of the trait between twin pairs. B. Parent-offspring regression. Measures the relationship between parents and offspring traits. Slope of regression line indicates narrow sense heritability H squared. C. Adoption studies. Compares adopted children with biological and adoptive parents. If a trait is highly heritable, adopted children should resemble their biological parents more than their adoptive parents. D. Genome-wide association studies GWAS. Identifies genetic variants associated with traits. Estimates SNP. Compares adopted children with biological and adoptive parents. If a trait is highly heritable, adopted children should resemble their biological parents more than their adoptive parents. D. Genome-wide association studies GWAS. Identifies genetic variants associated with traits. Estimates SNP-based heritability, which accounts for heritable variation due to known genetic markers. 4. Interpretation of heritability. Heritability is population-specific. It varies depending on genetic diversity and environmental factors. High heritability does not mean immutability. Even if a trait has high heritability, e.g., IQ or height, environmental factors, nutrition, education can still influence it. Heritability does not apply to individuals. It describes population-level variation, not the genetic contribution to an individual's trait. 5. Heritability of common traits in humans. Trait heritability estimate H squared, height approximately 0.8, 80% intelligence IQ, approximately 0.5 minus 0.7, 50 minus 70%, body mass index BMI, approximately 0.4 minus 0.7, 40 minus 70%, personality traits approximately 0.4 minus 0.5, 40 minus 50%, schizophrenia approximately 0.8, 80%. These values indicate the proportion of variation in these traits that is due to genetics, but they do not mean that the trait is purely genetic. 6. Solve previous year questions 1. Question. Which of the following statements about heritability is true? A. It describes the extent to which an individual's trait is inherited from parents. B. It varies depending on environmental conditions. C. It remains constant for a given trait across all populations. D. A trait with high heritability is not influenced by the environment. Answer. B. It varies depending on environmental conditions. Heritability depends on genetic and environmental variation within a population. 2. Question. If the heritability of height in a population is 80%, what does this mean? A. Each person inherits 80% of their height from their parents. B. 80% of an individual's height is determined by genes, while 20% is determined by the environment. C. 80% of the variation in height within this population is due to genetic differences. D. Height is completely determined by genetic factors. Answer. C. 80% of the variation in height within this population is due to genetic differences. Heritability refers to variation in a population, not individual inheritance. 7. Multiple choice questions MCQs for practice 1. Broad sense heritability H squared includes which of the following genetic components? A. Additive genetic variance. B. Dominance genetic variance. C. Epistatic genetic variance. C. All of the above. Answer. D. All of the above. 2. Which method is commonly used to estimate heritability in human traits? A. Test cross. B. Twin studies. C. Hardy-Weinberg equilibrium calculations. D. Southern blotting. Answer. B. Twin studies. 3. What does a narrow sense heritability value of 0.3 indicate? A. The trait is largely determined by environmental factors. B. 30% of the phenotypic variation is due to additive genetic effects. C. The trait is completely inherited. D. The trait does not respond to selection. Answer. B. 30% of the phenotypic variation is due to additive genetic effects. 8. Conclusion. Heritability is a fundamental concept in genetics that quantifies the genetic contribution to trait variation in a population. Understanding heritability helps in evolutionary biology, medicine, and selective breeding. However, it must be interpreted carefully, considering environmental influences and population specificity. You said, QTL mapping. ChatGPT said. QTL mapping, quantitative trait loci mapping 1. Introduction to QTL mapping. Quantitative trait loci QTL mapping is a statistical method used to identify the genetic regions loci associated with complex traits that show continuous variation, such as height, weight, disease susceptibility, and yield in crops. Unlike Mendelian traits controlled by a single gene, quantitative traits result from the combined effects of multiple genes and environmental factors. QTL mapping helps researchers locate these genetic regions and understand how they contribute to phenotypic variation. 2. Key concepts in QTL mapping. A quantitative trait loci QTL. A QTL is a genomic region that influences a quantitative trait. These loci contain one or more genes contributing to trait variation. QTLs do not necessarily represent single genes but genomic segments associated with the trait. B. Linkage mapping versus association mapping. Linkage mapping uses controlled crosses between genetically diverse parents to track how traits are inherited in offspring. Association mapping GWAS, genome-wide association study, uses natural populations to identify genetic variants associated with traits, relying on historical recombination events. 3. Steps in QTL mapping. Step 1. Development of mapping population. A QTL study begins with a genetically diverse population derived from controlled crosses. Common populations include F2 population, derived from crossing two inbred lines and allowing self-fertilization for one generation. Backcross BC, cross between an F1 hybrid and one of the parental lines. Recombinant inbred lines, RILs, created by repeated selfing or sibling crosses, leading to stable lines. Near-isogenic lines, NILs, differ only in specific genomic regions containing the QTL, making them ideal for fine mapping. Step 2. Genotyping the population. DNA markers, e.g., SNPs, SSRs, RFLPs, are used to track genetic variation. The goal is to identify genetic markers that segregate with the trait of interest. Step 3. Phenotyping the population. The trait of interest, e.g., plant height, disease resistance, blood pressure, is measured across individuals in the mapping population. Step 4. Statistical analysis to identify QTLs. Linkage analysis identifies markers co-segregating with the trait. LOD-score logarithm of odds determines the likelihood of a marker being linked to a QTL. A LOD score in GT3 is considered significant. Interval mapping uses statistical models to estimate the QTL's effect and location. Composite interval mapping (CIM) enhances detection by controlling background genetic variation. Step 5. Fine mapping and candidate gene identification. Once QTLs are identified, fine mapping is used to narrow down the specific genes involved. Advanced techniques like GWAS and CRISPR help validate candidate genes. 4. Applications of QTL mapping. Agriculture and plant breeding. Used to improve crop yield, drought resistance, disease resistance, and nutrient efficiency. Example, identification of QTLs for high-yield traits in rice and wheat. B. Medical and human genetics. Helps understand genetic basis of complex diseases such as diabetes, hypertension, and schizophrenia. Example, mapping QTLs for obesity and cardiovascular diseases. C. Evolutionary biology. Used to study genetic variation and adaptation in populations. Example, identification of QTLs involved in beak size evolution in Darwin's finches. 5. Solve previous year questions. 1. Question. What is the primary goal of QTL mapping? A. To identify single gene traits. B. To locate regions of the genome associated with complex traits. C. To determine the sequence of an entire genome. D. To study epigenetic modifications. Answer. B. To locate regions of the genome associated with complex traits. 2. Question. Which of the following markers is commonly used in QTL mapping? A. SNPs, single nucleotide polymorphisms. B. Transposons. C. Histone modifications. D. microRNAs. Answer. A. SNPs, single nucleotide polymorphisms. 6. Multiple choice questions MCQs for practice 1. Which of the following techniques is most commonly used in QTL mapping? A. PCR. B. Genome sequencing. C. Linkage analysis and interval mapping. D. Karyotyping. Answer. C. Linkage analysis and interval mapping. 2. What does a high LOD score in QTL analysis indicate? A. No linkage between marker and trait. B. Strong evidence for linkage. C. Recombination between marker and QTL. D. That the trait is completely determined by a single gene. Answer. B. Strong evidence link. 3. Which of the following is an advantage of QTL mapping? A. It identifies the exact causal gene for a trait. B. It can locate multiple genetic regions affecting a trait. C. It works best for single gene traits. D. It does not require a mapping population. Answer. B. It can locate multiple genetic regions affecting a trait. 7. Conclusion. QTL mapping is a powerful approach for identifying genetic loci associated with quantitative traits. It has broad applications in plant breeding, medical genetics, and evolutionary biology. By integrating modern techniques like GWAS and CRISPR, researchers can refine QTL studies to pinpoint genes responsible for complex traits, aiding in genetic improvement and disease research. You said. No table encodes from now on, only paragraph. ChatGPT said. Memory updated. Quantitative trait loci QTL mapping is a genetic approach used to identify the specific regions of the genome associated with quantitative traits, which are traits influenced by multiple genes and environmental factors. Unlike Mendelian traits controlled by single genes, quantitative traits, such as height, weight, and disease susceptibility, show continuous variation. QTL mapping is essential for understanding the genetic basis of complex traits and is widely used in agriculture, medicine, and evolutionary biology. The process of QTL mapping begins with the development of a mapping population. This population is derived from crosses between genetically diverse parents that show significant variation in the trait of interest. Common types of mapping populations include F2 populations, backcross populations, recombinant inbred lines, and near isogenic lines. Once the population is established, genotyping is performed using molecular markers such as single nucleotide polymorphisms, SNPs, and simple sequence repeats, SSRs, which help in tracking genetic variation across the genome. After genotyping, the next step is phenotyping, where the individuals in the mapping population are measured for the trait of interest under controlled conditions. The combination of genotypic and phenotypic data allows researchers to perform statistical analyses to identify regions of the genome linked to the trait. One of the key statistical methods used is linkage analysis, which determines whether a genetic marker is inherited together with the trait more frequently than expected by chance. The logarithm of odds (LOD) score is used to assess the strength of linkage between markers and QTLs, with higher scores indicating stronger evidence for the presence of a QTL. Several mapping approaches are used to improve the accuracy of QTL detection. Interval mapping estimates the location and effect of a QTL within a defined genomic region. Composite interval mapping refines this process by controlling for genetic background effects, making it easier to detect multiple QTLs with greater precision. Once QTLs are identified, fine mapping is conducted to narrow down the genetic region and identify candidate genes responsible for the trait. QTL mapping has significant applications in plant and animal breeding, as it helps breeders select individuals with favorable genetic traits for improved yield, disease resistance, and environmental adaptation. In human genetics, QTL mapping contributes to understanding the genetic basis of complex diseases such as diabetes, hypertension, and schizophrenia by identifying genomic regions associated with disease susceptibility. Evolutionary biologists also use QTL mapping to study genetic adaptation and variation in natural populations. One of the limitations of QTL mapping is that it requires a well-structured mapping population, which may not always be available for certain species, particularly humans. Additionally, QTL mapping often identifies large genomic regions containing multiple genes, making it challenging to pinpoint the exact causal genes. Advances in genome-wide association studies (GWAS) and next-generation sequencing have helped overcome some of these limitations by providing higher-resolution mapping of genetic variants. In summary, QTL mapping is a fundamental tool in genetics that helps identify genomic regions associated with complex traits. By integrating molecular markers, statistical analysis, and controlled breeding experiments, researchers can uncover the genetic basis of important traits in agriculture, medicine, and evolutionary biology. Despite its challenges, QTL mapping remains a powerful approach for studying quantitative traits and their underlying genetic architecture. You said. Pedigree analysis, ChatGPT said. Pedigree analysis is a method used in genetics to study the inheritance patterns of traits and diseases across multiple generations of a family. By analyzing pedigrees, researchers and geneticists can determine whether a trait follows Mendelian inheritance patterns, such as autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or mitochondrial inheritance. This approach is particularly useful in medical genetics for identifying hereditary disorders, understanding genetic risks, and guiding genetic counseling. A pedigree is a graphical representation of a family tree, with standardized symbols used to denote individuals, relationships, and traits. Males are represented by squares, females by circles, and affected individuals are shaded. Horizontal lines indicate mating relationships, and vertical lines connect parents to their offspring. Pedigree charts allow researchers to trace the transmission of traits and identify inheritance patterns based on the recurrence of a condition in different generations. In autosomal dominant inheritance, a trait appears in every generation, and affected individuals usually have at least one affected parent. Both males and females are equally affected, and there is a 50% chance of passing the trait to offspring if one parent is affected. Examples of autosomal dominant disorders include Huntington's disease and Marfan syndrome. In contrast, autosomal recessive inheritance occurs when 2 copies of a mutant allele are necessary for an individual to express the trait. Unaffected carrier parents can pass the trait to their children, with a 25% chance of an affected child when both parents are carriers. Cystic fibrosis and sickle cell anemia follow this pattern. X-linked X-linked dominant inheritance involves genes located on the X chromosome. Affected males transmit the trait to all their daughters but not to their sons, while affected females can pass the trait to both sons and daughters. X-linked dominant inheritance involves genes located on the X chromosome. Affected males transmit the trait to all their daughters but not to their sons, while affected females can pass the trait to both sons and daughters. Examples include Rett syndrome and some forms of vitamin D-resistant rickets. In X-linked recessive inheritance, males are more frequently affected because they inherit only one X chromosome from their mother. Carrier females rarely show symptoms but can pass the affected allele to their sons. Hemophilia and Duchenne muscular dystrophy are classic examples of X-linked recessive disorders. Mitochondrial inheritance follows a maternal pattern since mitochondria, and their DNA are inherited exclusively from the mother. All offspring of an affected mother inherit the condition, whereas affected fathers do not pass the trait to their children. Disorders such as Leber's hereditary optic neuropathy and mitochondrial myopathy follow this inheritance pattern. Pedigree analysis is valuable for predicting genetic risks, diagnosing inherited disorders, and assisting in genetic counseling. By understanding inheritance patterns, clinicians can provide families with information about recurrence risks, carrier testing, and possible interventions. Advances in molecular genetics, such as whole genome sequencing, complement traditional pedigree analysis by confirming genetic diagnoses and identifying novel mutations responsible for inherited conditions. Despite its limitations and complex traits influenced by multiple genes and environmental factors, pedigree analysis remains an essential tool in medical and evolutionary genetics. You said, karyotyping, ChatGPT said. Karyotyping is a laboratory technique used to analyze the number, size, shape, and structure of chromosomes in an organism's cells. It is a fundamental tool in genetics for detecting chromosomal abnormalities associated with genetic disorders, congenital anomalies, infertility, and certain cancers. By arranging chromosomes in a standardized format based on their size, banding patterns, and centromere position, karyotyping allows geneticists to identify numerical and structural chromosomal variations. The process of karyotyping begins with the collection of cells from sources such as blood, bone marrow, amniotic fluid, or chorionic villus samples. These cells are then cultured in a growth medium to promote cell division, as chromosomes are most visible during the metaphase stage of mitosis. Chemical agents such as colchicine is used to arrest cells in metaphase, ensuring that chromosomes are condensed in line for microscopic examination. The cells are then treated with a hypotonic solution to spread the chromosomes, followed by fixation and staining with dyes like DIMSA, which highlights specific banding patterns unique to each chromosome. There are different types of karyotyping techniques based on staining methods. G-banding, the most commonly used method, produces a characteristic pattern of dark and light bands that help distinguish individual chromosomes. Other techniques include Q-banding, which uses quinacrine fluorescence, and R-banding, which enhances the visibility of certain regions by reversing the staining pattern of G-banding. High-resolution karyotyping allows for the detection of subtle chromosomal changes by analyzing chromosomes at an earlier stage of mitosis when they are less condensed. Karyotyping is primarily used to diagnose chromosomal disorders. Numerical abnormalities involve changes in chromosome number, such as trisomy 21, Down syndrome, where an extra copy of chromosome 21 is present. Other examples include Turner syndrome 45, X where one X chromosome is missing in females, and Klinefelter syndrome 47, XXY, where males have an extra X chromosome. Structural abnormalities include deletions, duplications, inversions, and translocations, which can lead to genetic disorders such as Cri-du-Chat syndrome, caused by a deletion on chromosome 5, and chronic myeloid leukemia, associated with the Philadelphia chromosome translocation between chromosomes 9 and 22. In prenatal diagnostics, karyotyping is widely used for detecting chromosomal abnormalities in fetuses through procedures like amniocentesis and chorionic villus sampling. This allows for early identification of genetic disorders, enabling informed medical decisions and counseling for parents. In oncology, karyotyping helps in identifying chromosomal alterations in cancer cells, aiding in diagnosis, prognosis, and treatment planning. Despite its usefulness, karyotyping has limitations in detecting smaller genetic mutations, such as single-gene disorders or microdeletions, which require more advanced molecular techniques like fluorescence in situ hybridization FISH and chromosomal microarray analysis CMA. However, karyotyping remains a crucial and widely used cytogenetic technique for diagnosing chromosomal abnormalities and understanding genetic disorders. You said. Genetic disorders Downs ChatGPT said. Genetic disorders are conditions caused by abnormalities in an individual's DNA, which can result from mutations in a single gene, chromosomal alterations, or complex interactions between multiple genes and environmental factors. One of the most well-known genetic disorders is Down syndrome, also known as trisomy 21, which is caused by the presence of an extra copy of chromosome 21. This condition leads to characteristic physical features, intellectual disabilities, and an increased risk of various health complications. Down syndrome occurs due to an error in cell division known as non-disjunction, where chromosome 21 fails to separate properly during gamete formation. As a result, an individual inherits 3 copies of chromosome 21 instead of the usual 2. There are 3 main types of Down syndrome: trisomy 21, which accounts for about 95% of cases and results from a full extra chromosome 21 in every cell; mosaic Down syndrome, where only some cells have the extra chromosome, leading to milder symptoms; and translocation Down syndrome, where a piece of chromosome 21 attaches to another chromosome, usually chromosome 14, due to a structural rearrangement. The clinical features of Down syndrome include distinct facial characteristics such as a flat facial profile, upward slanting eyes, a small nose, and a protruding tongue. Affected individuals often have hypotonia, low muscle tone, short stature. Down syndrome occurs due to an error in cell division known as non-disjunction, where chromosome 21 fails to separate properly during gamete formation. As a result, an individual inherits 3 copies of genetic disorders. Down's ChatGPT said. Genetic disorders are conditions caused by abnormalities in an individual's DNA, which can result from mutations in a single gene, chromosomal alterations, or complex interactions between multiple genes and environmental factors. One of the most well-known genetic disorders is Down syndrome, also known as trisomy 21, which is caused by the presence of an extra copy of chromosome 21. This condition leads to characteristic physical features, intellectual disabilities, and an increased risk of various health complications. Down syndrome occurs due to an error in cell division known as non-disjunction, where chromosome 21 fails to separate properly during gamete formation. As a result, an individual inherits 3 copies of chromosome 21 instead of the usual 2. There are 3 main types of Down syndrome: trisomy 21, which accounts for about 95% of cases and results from a full extra chromosome 21 in every cell; mosaic Down syndrome, where only some cells have the extra chromosome, leading to milder symptoms; and translocation Down syndrome, where a piece of chromosome 21 attaches to another chromosome, usually chromosome 14. There are 3 main types of Down syndrome: trisomy 21, which accounts for about 95% of cases and results from a full extra chromosome 21 in every cell; mosaic Down syndrome, where only some cells have the extra chromosome, leading to milder symptoms; and translocation Down syndrome, where a piece of chromosome 21 attaches to another chromosome, usually chromosome 14, due to a structural rearrangement. The clinical features of Down syndrome include distinct facial characteristics such as a flat facial profile, upward slanting eyes, a small nose, and a protruding tongue. Affected individuals often have hypotonia, low muscle tone, short stature, and a single palmar crease in the hand. Cognitive impairments vary, with most individuals experiencing mild to moderate intellectual disability and delayed developmental milestones. Down syndrome is also associated with congenital heart defects, gastrointestinal abnormalities, hearing loss, vision problems, and an increased risk of thyroid dysfunction and leukemia. Prenatal screening and diagnostic tests are available to detect Down syndrome before birth. Screening tests, such as maternal serum markers and non-invasive prenatal testing (NIPT), assess the likelihood of the condition based on biochemical and DNA analysis of fetal cells in maternal blood. Confirmatory diagnostic tests, including chorionic villus sampling (CVS) and amniocentesis, provide definitive genetic information but carry a small risk of miscarriage. There is no cure for Down syndrome, but early intervention programs, medical management, and supportive therapies improve the quality of life for affected individuals. Specialized education, speech therapy, occupational therapy, and physical therapy help address developmental challenges, while regular medical checkups ensure early detection and treatment of associated health conditions. Advances in medical care have significantly increased the life expectancy of individuals with Down syndrome, with many living into their 60s and leading fulfilling lives with appropriate support. Research on Down syndrome continues to focus on understanding its genetic basis, improving medical treatments, and enhancing cognitive and developmental outcomes. Studies exploring gene expression, potential drug therapies, and the role of neuroplasticity offer hope for future interventions that may mitigate the effects of the extra chromosome 21. You said, Down in detail. ChatGPT said. Down syndrome, a detailed overview. Introduction. Down syndrome, also known as trisomy 21, is a genetic disorder caused by the presence of an extra copy of chromosome 21. It is one of the most common chromosomal disorders, affecting approximately 1 in 700 live births worldwide. The condition is characterized by distinct physical features, developmental delays, intellectual disabilities, and an increased risk of certain medical conditions. While there is no cure, early intervention, medical care, and supportive therapy significantly improve the quality of life for individuals with Down syndrome. Causes and genetic basis. Down syndrome results from an error in chromosome segregation during cell division, leading to an extra chromosome 21. This abnormality occurs due to non-disjunction, translocation, or mosaicism. Trisomy 21, non-disjunction Down syndrome. This is the most common form, accounting for about 95% of cases. It occurs when chromosome 21 fails to separate properly during the formation of egg or sperm cells meiosis. As a result, the fertilized egg ends up with 3 copies of chromosome 21 instead of the usual 2. Translocation Down syndrome accounts for about 3–4% of cases. Occurs when an extra portion of chromosome 21 attaches to another chromosome, often chromosome 14, instead of being a separate extra chromosome. Unlike trisomy 21, this form can be inherited from a parent carrying a balanced translocation, meaning that they do not have Down syndrome but can pass a rearranged chromosome to their children. Mosaic Down syndrome. The rarest form, occurring in about 1–2% of cases. Happens when some cells in the body have a normal 46 chromosomes, while others have an extra chromosome 21. Since not all cells are affected, individuals with mosaic Down syndrome may have milder symptoms compared to those with full trisomy 21. Clinical features and symptoms. Individuals with Down syndrome have a combination of characteristic physical features, developmental delays, and medical conditions. 1. Physical features. Flat facial profile and small nose, upward slanting eyes with epicanthic folds, small ears and a short neck, protruding tongue due to a small oral cavity, hypotonia, low muscle tone leading to delayed motor development, short stature and shorter limbs. Single transverse palmar crease in the hand, small hands and feet with a wide gap between the first and second toes. 2. Intellectual and developmental characteristics. Intellectual disability ranging from mild to moderate, delayed speech and language development, slower cognitive processing and learning ability, short attention span and difficulty with problem-solving, delayed motor skills such as sitting, standing, and walking. 3. Medical conditions associated with Down syndrome. Congenital heart defects, present in nearly 50% of affected individuals, including atrioventricular septal defect (AVSD) and ventricular septal defect (VSD). Gastrointestinal abnormalities, conditions like duodenal atresia, Hirschsprung disease, and gastroesophageal reflux are more common. Hearing and vision problems, higher risk of conductive hearing loss and vision disorders such as cataracts and strabismus. Thyroid dysfunction, hypothyroidism is frequently observed. Leukemia, increased risk of acute lymphoblastic leukemia (ALL) and acute myeloid leukemia (AML) in childhood. Obstructive sleep apnea, due to narrowed airways and muscle hypotonia. Alzheimer's disease, early onset dementia is more common due to the overexpression of amyloid precursor protein (AP) on chromosome 21. Diagnosis. Down syndrome can be diagnosed prenatally or after birth through a combination of screening and diagnostic tests. 1. Prenatal screening tests estimate the likelihood of Down syndrome but do not confirm it. First trimester screening measures levels of pregnancy-associated plasma protein A (PAP-A) and human chorionic gonadotropin (HCG) along with an ultrasound to check for nuchal translucency. Second trimester screening quad test measures levels of AFP, HCG, estriol, and inhibin A in maternal blood. Non-invasive prenatal testing (NIPT) analyzes fetal DNA and maternal blood for chromosomal abnormalities. 2. Diagnostic tests definitive but invasive. Chorionic villus sampling (CVS) performed between 10 to 13 weeks of pregnancy, involving the extraction of placental tissue for chromosomal analysis. Amniocentesis, conducted between 15 to 20 weeks, where amniotic fluid is analyzed for fetal chromosomes. 3. Postnatal diagnosis. Physical examination, based on characteristic features. Karyotyping, confirms the presence of an extra chromosome 21. Management and treatment. Although there is no cure for Down syndrome, early intervention, medical care, and support can improve the quality of life for affected individuals. 1. Early intervention and education. Special education programs tailored to individual learning needs. Speech therapy, occupational therapy, and physical therapy to enhance communication and motor skills. 2. Medical management. Regular checkups with a pediatrician or genetic specialist. Cardiac evaluations for congenital heart defects. Routine thyroid function tests and hearing screenings. Vision assessments and corrective treatments for eye disorders. Nutritional support to maintain a healthy weight. 3. Social and emotional support. Support groups for families and caregivers. Encouragement of social inclusion and participation in community activities. Life skills training to promote independence. 4. Research and future treatments. Studies exploring gene therapy and potential drugs to enhance cognitive function. Advances in regenerative medicine and neuroplasticity research. Ongoing improvements in prenatal diagnostics for early detection. Prognosis and life expectancy. Due to advances in medical care, individuals with Down syndrome now have a significantly improved life expectancy, averaging around 60 years. With proper support, many individuals can lead fulfilling lives, participate in education and employment, and engage in social activities. While challenges exist, societal awareness, inclusivity, and medical advancements continue to enhance opportunities for people with Down syndrome, promoting their independence and well-being. You said, Klinefelter's ChatGPT said. Klinefelter syndrome, a detailed overview introduction. Klinefelter syndrome (KS) is a genetic disorder that affects males and is caused by the presence of an extra X chromosome, resulting in a karyotype of 47, XXY instead of the typical 46, XY. It is one of the most common chromosomal disorders in males, with an estimated occurrence of 1 in 500 to 1,000 live male births. The condition affects physical, cognitive, and reproductive development, leading to symptoms such as small testes, infertility, reduced testosterone levels, gynecomastia, breast enlargement, and learning difficulties. Despite these challenges, early diagnosis and management, including testosterone therapy and educational support, can significantly improve quality of life. Causes and genetic basis. Klinefelter syndrome occurs due to a chromosomal non-disjunction error during the formation of sperm or egg cells, leading to an extra X chromosome. This results in a 47, XXY karyotype instead of the normal 46, XY male chromosome complement. In some cases, additional X chromosomes e.g., 48, XXXY or 49, XXXY can be present, leading to more severe symptoms. There are 3 main genetic variations of Klinefelter syndrome. Classic Klinefelter syndrome 47, XXY. The most common form, present in about 85–90% of cases. The extra X chromosome is present in all body cells. Mosaic Klinefelter syndrome 46, XY 47, XXY. Occurs when some cells have the extra X chromosome while others have the normal male karyotype. Symptoms tend to be milder, and fertility may be less severely affected. Variants with multiple X chromosomes 48, XXXY or 49, XXXY. These rare forms lead to more pronounced intellectual disabilities and physical abnormalities. The X chromosome disrupts normal male development by interfering with testosterone production, leading to the characteristic features of the syndrome. Clinical features and symptoms. The symptoms of Klinefelter syndrome vary widely, and some individuals may have mild or no noticeable signs. The most prominent effects relate to reproductive health, physical development, and cognitive abilities. 1. Physical characteristics. Small, firm testes, testicular atrophy, leading to reduced testosterone production. Gynecomastia, breast enlargement due to hormonal imbalance. Tall stature with long legs and a disproportionately shorter torso. Reduced facial and body hair compared to typical males. Decreased muscle mass and strength. Low bone density, increasing the risk of osteoporosis. Narrow shoulders and wider hips, giving a more feminized body shape. 2. Reproductive and endocrine features. Primary hypogonadism, failure of the testes to produce sufficient testosterone. Infertility due to low sperm production, azoospermia or oligospermia. Reduced libido and erectile dysfunction in some cases. Increased risk of testicular cancer. 3. Cognitive and behavioral characteristics. Mild to moderate learning disabilities, particularly in language and reading. Delayed speech and language development in childhood. Difficulty with executive functioning, such as organization and problem solving. Lower attention span and increased risk of attention deficit hyperactivity disorder (ADHD). Social difficulties, including shyness and social anxiety. 4. Associated health risks. Increased risk of metabolic disorders such as type 2 diabetes. Higher likelihood of developing autoimmune diseases like lupus and rheumatoid arthritis. Increased risk of cardiovascular disease and deep vein thrombosis. Thyroid dysfunction, particularly hypothyroidism. Greater susceptibility to breast cancer due to hormonal imbalances. Diagnosis. Klinefelter syndrome is often underdiagnosed due to its variable presentation. Many cases are not identified until adulthood, especially when individuals seek medical help for infertility. 1. Prenatal diagnosis. Can be detected through amniocentesis or chorionic villus sampling (CVS) during pregnancy if chromosomal analysis is performed. Non-invasive prenatal testing (NIPT) can also indicate the presence of an extra X chromosome. 2. Postnatal diagnosis. Physical examination. Doctors may suspect Klinefelter syndrome based on small testes, gynecomastia, and tall stature. Karyotyping, a blood test that confirms the presence of an extra X chromosome. Hormonal testing. Blood tests show low testosterone levels, elevated follicle-stimulating hormone (FSH), and luteinizing hormone (LH), indicating testicular dysfunction. Semen analysis reveals low or absent sperm count, confirming infertility in affected individuals. Management and treatment. There is no cure for Klinefelter syndrome, but various treatments and interventions can help manage symptoms and improve quality of life.